go to classics.com, click on practice. You will see packages. You can select the package you want. Since it's NEET classes, I'm selecting NEET Premium. Here you find tests in physics, chemistry, biology and NEET full test. Since we are doing 11th biology, I click on that. Each bubble here is a test. Now if you look at the green bubble, it means that the test has been completed. But if you really want to revisit the test again for the revision, any number of times you can take up all these tests. Now blue bubble indicates that you are not started the test. Yellow bubble indicates that you have not completed the test. You might have started the test. After 3 or 4 questions, you would have realized you are not really thorough with the concepts, and so you would be walking out of the test. Maybe later you will learn the concepts and then it indicates that you need to complete the test. And if you look at all the chapters here, every chapter has a different number of tests depending upon the complexity of the textbook and also its importance in the NEET exams. And so the tests are created, a different number of tests are created for different chapters. Here you find the randomized test in the red bubble. Once you have completed all these levels, you can just take up the randomized level where the complexity levels. I'm Hima Rajesh, tutor for biology. Today I will be covering excretory products and their elimination. So you just click on the practice button. After going to the classics.com page, you will find a lot of tests here. Chapter wise tests are here, and here if you look at it, each bubble is a test. If you have seen a green bubble, it means you have completed the test. But if you want to revisit the test for revision purpose, or if you have not got some of the answers you might have got wrong, in order to check whether you are able to recollect and answer, you can take the test again. Blue bubble indicates you are yet to take the test. Yellow bubble indicates you have started the test but you have not completed it. It means to say that you have might have started the test. After 3 or 4 questions, you might have realized you are not really thorough with the concepts, and so you would have just left the test. After learning the concept, it is an indicator that you need to take the test again and you are yet to complete the test. Now this red bubble is a randomized test. Here, after you complete all the individual tests, if you still want to check whether you are able to recollect randomly, then you can do this test. And if you look at it here, for all the chapters we have tests, and number of tests are varied depending upon the complexity of the chapter and also its importance in the NEET examination. Now here you have a revision button, notes for the button here. If you click it, it will open into NEET revision. Now I hope you have got to know how to navigate through the website. In today's class, I will be revising key concepts, do a practice test based on the concepts learned, and in the last, I'm going to answer your questions. Now, before we begin into the lesson, I'm sure since you're all awaiting for your NEET exams, there will be some amount of fear and tension. Basically, keep relaxed. Now, if you look at it, especially in biology, we have found that from our analysis that there are 3 kinds of questions. One is NCERT based. Approximately 68 to 70 questions in the examination are done directly from the NCERT books. They can be attempted very easily if you are thorough with your NCERT syllabus. Direct from the previous year's question papers, another 8 to 10 questions will feature, and if you have solved the past papers, you can easily score the marks even in that section. And modified questions, if you say 8 to 10 questions will be definitely new in the NEET exams. They would not have appeared earlier, and those questions become the game changer. These questions cannot be just answered by memorizing or mugging up the facts. Though they are from NCERT book, to get them right, you need to apply the knowledge that you have about the subject. So in this website, we have made an earnest attempt to help you prepare adequately for your NEET with the high scores. I hope you have now got the framework. Now let us begin the lesson. So I would just suggest you that it is ideal if you are able to have this package as a part of your study material. Now let us start with the revision notes. So excretory products and their elimination. Again, this PDF we have done for 11th and 12th physics, chemistry and biology to a tune of 135 pages, so that you are able to understand, you are able to revise very quickly, especially during the exam, when the examination time, a day before your exam you want to revise. It's impossible for you to revise looking into all the books of NCERT. So this would be an ideal material for you to revise, and you can add materials. Now let us get into excretory products and their elimination. Now, modes of excretion. The process of removal of nitrogenous waste from the body is called excretion. The organs of excretion are called excretory organs. There are different types of excretory products or waste materials. Different animals expel different nitrogenous waste. Now, what are the three modes of excretion? Let us have a look. First is ammonotelism. Main nitrogenous end product is ammonia. Ammonotelic excretion is found in aquatic plants like protozoans, sponges, coelentrates, aquatic arthropods, and mostly with aquatic mollusks, bony fishes, and frog tadpole. Ammonia is highly toxic because of its high pH. Ammonia is highly soluble in water, so large amount of water is required for its excretion. It is lost to general body surface into the surrounding water. Now that is why it is most commonly found in the aquatic animals. Now ureotelism. The main nitrogenous waste is the urea. Ureotelic excretion is found in those animals which can take in water and can retain considerable amount of urea in their blood. It is a common method of excretion. Sorry, there are some network issues and it is just going this way and that way. The main nitrogenous waste in ureotelism is urea. Ureotelic excretion is found in those animals which can take in water and can retain considerable blood. It's a common method of excretion in humans, whales, seals, camels, kangaroo, toads, frogs, sharks, etc. Please remember, frogs it is ureotelism, in tadpoles it is ammonotelism. Urea formation in liver cells of mammals was studied by Krebs and Henseleit. Urea is formed in the liver by detoxification of ammonia. Urea is transported in the body by blood plasma. A person who takes large amount of protein in his diet will excrete more urea. A person having more food, water will also have more urea in his blood. Less water is required for excretion. The main nitrogenous waste is crystals of uric acid. It is common in birds, land reptiles, insects, snails, etc. Uric acid is least soluble in water and it is less toxic. It is excreted in solid crystal forms formed from ammonia in the liver of uricotelic animals and malpighian tubules in the insects. Now let us look at the excretory system of man. So you know kidney is the main excretory organ. So what does the excretory system as a whole consist of? Excretory system of human consists of following parts: a pair of kidneys, a pair of ureters, one urinary bladder, and one urethra. Now, kidneys are bean-shaped structures. They are chocolate brown structures lying in the abdomen, one on each side of the vertebral column, just below the diaphragm. The left kidney is higher than the right kidney, but it is reversed in the rabbit. Concavity of kidney, called hilum or hilus, is always directed inwards. These form the urine and control the osmotic pressure within the organism with respect to external environment. Now let us look at the structure of this is the structure of a single kidney. Here you are able to see it. The blood vessels, ureter and nerves enter the kidney through the hilum. Outer layer is a tough capsule and outer layer or the zone is called as the cortex and inner layer is called as the medulla. The medulla, you will be seeing medullary pyramids which are getting into the calyces. Cortex extends between medullary pyramids called columns of Bertini. So this is the structure, and look at the diagram and learn the parts. They might ask you to associate the parts. Now structure of the nephron. In the structure, if you look at nephron, it is the structural and functional unit of kidney. These are also called as renal tubules or uriniferous tubules. There are about 1 million nephrons in each kidney. Each nephron is about 6 centimeters long. It is differentiated into 4 regions. Bowman's capsule. So this is the Bowman's capsule. It is a double-walled cup and it is lined by thin flat cells called podocytes. It contains group of capillaries called glomerulus. Glomerulus in the kidney acts as a dialyzing bag. So the glomerulus within it will act as the dialyzing bag and it is the filtering unit. Now next look at the proximal convoluted tubule. It is a highly coiled or convoluted tubular structure. It is about 12 to 24 millimeters in length. Almost whole of vitamins, glucose, amino acids, sodium and potassium, etc. are reabsorbed by active transport. The next is the Henle's loop or loop of Henle. It is a U-shaped segment. Loop of Henle is long in mammals and birds which secrete hypertonic urine, but short in other vertebrates like reptiles. Now, distal convoluted tube, DCT. It is connected to the collecting duct. Active reabsorption of some sodium takes place. It is impermeable to water. Now you look at the ureters. Two narrow tubes arising from the hilum are called ureters. Each is about 30 centimeters in length and these bring the urine downwards and open into urinary bladder. Urinary bladder, that is, each ureter will open into a bag-like structure and it is called as the urinary bladder. It temporarily stores the urine. It can hold about 0.5 to 1 liters of urine. It is absent in birds. In both reptiles and birds, ureters and rectum open into a common sac called the cloaca. Urethra. It's a muscular and tubular structure which extends from the neck of the bladder to the outside. In females, this tube is small and serves as a passage of urine only. In males, it is long and functions as a common passage for urine and spermatic fluids. Now, with this understanding of the nephrons, you have two kinds of nephrons. One is cortical nephron, the other is juxtamedullary nephrons. Cortical nephrons are in majority of the nephrons, loop of Henle is too short and extends very little into the medulla, and these are called as the cortical nephrons. In juxtamedullary nephrons, loop of Henle will be very long and it runs deep into the medulla. Now let us look into the urine formation. So there are 3 steps in the urine formation. One is glomerular filtration, second is reabsorption, and third is tubular secretion. Now let us look into glomerular filtration. The glomerular capillary blood pressure causes filtration of the blood through 3 layers, that is in the endothelium of the glomerular blood vessels, epithelium of Bowman's capsule, and the basement membrane between these 2 layers. Epithelial cells of Bowman's capsule called podocytes are arranged in an intricate manner so as to leave some minute spaces called filtration slits or slit pores. Blood is filtered very finely through these membranes and it is also considered to be ultrafiltration. The amount of filtrate formed by the kidney per minute is called glomerular filtration rate or GFR and it is about 125 ml per minute, that is 180 liters per day. Juxtaglomerular apparatus is a sensitive region formed by cellular modifications in the distal convoluted tubule and the afferent arteriole. A fall in GFR can activate juxtaglomerular cells to release renin, which can stimulate the glomerular blood flow and thereby the GFR comes back to normal. This I will be explaining in a few minutes from now. The net driving force which is responsible for ultrafiltration through glomerular blood capillaries and Bowman's capsule is provided by the difference between the hydrostatic pressure at which the blood enters the glomerulus and the sum of those pressures which resist the hydrostatic pressure. Now let us look into the next step, that is reabsorption. About 99% of the filtrate is usually reabsorbed by the renal tubules, and this is called reabsorption. Tubular epithelial cells in the nephron carry out this by active mechanisms, carry out by passive and active mechanisms. For example, glucose, amino acids, sodium, etc. are reabsorbed actively, and nitrogenous wastes are absorbed by passive transport. Third is tubular secretion. In tubular secretion is opposite to tubular reabsorption. Most substances that enter the tubule by tubular secretion move by active transport. It plays a minor role in function of human kidneys, but in animals like marine fishes and desert and amphibians, most nephrons do not have developed glomeruli. Their urine formed is maintained by the tubular secretion of urea, creatinine, and mineral ions. Now let us look into the composition of the urine. Now when we are looking at the composition of the urine, it is a pale yellow colored fluid due to the presence of the urochrome pigment. It is acidic in nature and it is slightly heavier than water. It has a faint aromatic odor due to urinate. Daily urine output in normal adult is 1.5 to 1.8 liters. It consists of water to an extent of 95 to 96%. Urea will be to an extent of 2%, and salt as sodium chloride will be about 2%, and other substances like uric acid, creatinine, etc. are 0.4%. Now let us look into the regulation of kidney function. Before that, let me talk about countercurrent mechanism. Countercurrent mechanism is to counteract because the outflow runs parallel to and in the opposite direction of the inflow. That is, there is ascending limb is there and descending limb is there. The flow will be in the opposite direction, and that is why we have this countercurrent mechanism. So, NaCl is transported from the ascending limb of the Henle's loop to the descending limb of vasa recta. The ascending limb of vasa recta in turn transports NaCl to the interstitium. Interstitium is the tissue between Henle's loop and vasa recta. Thus, a concentration gradient of 300 millimeter in the cortex to 1200 milliosmoles per liter in the medulla is created. Urea contributes to the process by being transported by ascending limb of the loop of Henle to the interstitium. As urine flows downwards in the collecting tubule, it encounters higher and higher concentration of the solutes in the interstitium. Hence, it goes on losing water due to exosmosis, and thus urine gets concentrated. Now, let us look about how osmoregulation occurs. How is the kidney function regulated? When there is an increased osmotic pressure of the blood. So, when there is increased blood pressure of the blood, what happens? Osmotic pressure, pituitary gland gets stimulated. When pituitary gland is getting stimulated, that is antidiuretic hormone or ADH is released in excess. ADH raise results in water permeability in DCT and CT. Concentrated urine is produced in less volume and osmotic pressure of the blood is reduced, that is, it strikes to normal. Similarly, when there is a decreased osmotic pressure of blood, osmoreceptors get depressed in the hypothalamus, and this results in inhibition of pituitary glands. When pituitary glands are inhibited, ADH secretion becomes lesser, so water permeability reduces in DCT and CT, as a result of which excess urine, which is hypotonic, is produced and osmotic pressure of the blood rises. Now let us look into osmoregulation and regulation of kidney functions. In humans, kidneys play an important role in osmoregulation. Osmotic pressure of the blood influences many cellular activities. Therefore, the amount of water in the body fluids must be regulated. In case the volume of body fluids fall below the normal, glomerular filtration slows down due to decrease in the blood pressure and filtration pressure in the glomerular capillaries. This stimulates the posterior pituitary lobe to release ADH. ADH increases the reabsorption of water in the DCT and the collecting duct. Less filtration and more reabsorption of water will lead to a discharge of small amount of hypotonic urine. This raises the volume of the body fluids to the normal. Reabsorption of sodium is controlled by a hormone aldosterone produced by the cortex of adrenal glands. A reduction in the level of sodium ion stimulate juxtaglomerular apparatus cells to form a protein called renin. Renin acts as an enzyme which converts plasma protein angiotensinogen into angiotensin. Reabsorption of sodium ions brings about the uptake of an osmotically equivalent amount of water. Absorption of sodium and water increases the blood volume and pressure. Thus, renin, angiotensin, aldosterone system. This we call it as RAS, R-A-A-S, RAS system, which maintains homeostasis. Thus, kidneys regulate body fluid osmoregulation by controlling the amount of water with the help of hormone ADH and sodium with the help of aldosterone and proteins by renin and angiotensin. Now, next, the urine is formed. It has to be expelled from the body. So, expulsion of the urine is known as micturition, and it is from the urinary bladder. It is a reflex process but can be controlled voluntarily to some extent in grown-up children and adults. Now, if you look at a newborn baby, it does not have control over the bladder, so it just urinates. And similarly, in adults, if you see like old people, aged people, they also would have lost control over the bladder, and so they can't withstand urine for a sense the signals, which causes the stretching of the urinary bladder when it gets filled with urine. In response, the stretch receptors on the walls of the bladder send signals to the CNS. CNS passes on the motor message to initiate contraction of smooth muscles of the bladder and simultaneous relaxation of the urethral sphincter, causing the release of urine. An adult human excretes on an average 1 to 1.5 liters of urine per day. On an average, 25 to 30 grams of urine is excreted out per day. Now with this, let us look into the last part. Now what are the other organs that help in excretion? So you know we have lungs, lungs remove about 18 liters per day of carbon dioxide and major quantities of water every day. Liver secretes bilirubin, bilirubinemia also help in excreting cholesterol, degraded steroid hormones, vitamins and drugs which pass out along with digestive water. Skin eliminates certain waste with sweat such as NaCl, some urea, lactic acid, etc. Sebaceous glands in the skin eliminate substances like sterols, hydrocarbons and waxes through sebum. Now let us look into the disorders of the urinary system. First is diabetes insipidus. It's a kind of diluted urine with reduction of fluid intake having no effect on the concentration of urine. The common diabetes insipidus is caused by the deficiency of arginine vasopressin, which is also called as AVP, also known as antidiuretic hormone. See, there are two diabetes which you should be aware. One is diabetes insipidus, which is due to deficiency of ADH, and this is an excretory system disorder. Other is diabetes mellitus, which is due to the deficiency of insulin. Now, uremia. It is the presence of an excessive amount of urine in the blood. It results from the decreased secretion of urea in the kidney tube, bacterial infection or some mechanical obstruction. Urea poisons the cells at high concentration. High concentration of non-protein nitrogen like urea, uric acid, creatinine exist in the blood due to kidney failure. Renal failure. The partial or total inability of the kidneys to carry out excretory function is called renal kidney failure. It is caused by tubular injury, bacterial toxins or inflammations, etc. Sometimes excess of alcohol can also result in renal failures. Excess of hormonal imbalance, uric acid formation, milk intake, dehydration, metabolic disturbances can lead to the formation of renal stones or villi. Nephritis, it's an inflammation of the renal pelvis, calluses and interstitial tissue due to local bacterial inflammation. Inflammation affects the countercurrent mechanism and the victim fails to produce the concentrated urine. Symptoms are back pain and frequent and painful urination. So there are certain other kinds of disorders like blood sugar. This will be a result of glucose in the urine due to diabetes mellitus. Ketonuria is urine with ketonic bodies like acetoacetic acid, beta hydroxyl butyric acid. Albuminuria is with albumin proteins in conditions like nephritis. Jaundice, wherein bipigments will be passed through the urine. Hemoglobinuria, presence of hemoglobin in urea. Pyuria, that is presence of pus cells in urea. Alkaptonuria, where there is a presence of a substance called alkapton in urine, which turns black when urine comes in contact with air, and it is called black urine disease. It's genetic and an inborn disease. Now with this, we have completed the key concepts that are important from NEET point of view. Now let us go and take a practice test. So this is inside the kidney. There are two zones, and dash and dash. The medulla is divided into few conical masses projecting into the dash. The cortex extends in between the medullary pyramids, and this is given. All this, if you are able to identify A, B, C, D, and E, you will be able to get the answer. Now let us look at the answer. Now what is A is the outer cortex, B is the inner medulla, C is medullary pyramids, D is the calyces, and then we are supposed to identify E. Where is E? This is E. This is the renal columns. So let us look into the choice. One is outer cortex. So this is the outer cortex, inner medulla, medullary pyramids, calyces, renal columns. So this is the answer. Identify F, G, H, I and J in order. So what is F? F is renal capsule, G is renal artery, H is renal vein, I is renal pelvis, J is ureter. Now let us look into the renal capsule, renal artery, renal pelvis, renal capsule, renal artery, renal vein, renal pelvis, and ureter. So this is the choice. Each nephron has two parts, the glomerulus and the renal tubule. The renal tubule begins with that and continues further to form a highly coiled network. B. C is the next part of the tubule whose ascending limb continues as highly coiled tubular region called dash. The F of many nephrons open into a straight tube called dash. So if you look at it, it is very clear. It is Bowman's capsule, proximal convoluted tubule, Henle's loop, distal convoluted tubule, distal convoluted tubules, and the collecting ducts. Sorry. Each nephron has got two parts, the glomerulus. Glomerulus is a tuft of capillaries formed by the dash. Blood from the glomerulus is carried by and dash. The renal tubule begins with dash which encloses the glomerulus along with dash is called the dash. Tubule continues further to form a highly coiled network. So if we are looking at this diagram, it has to be, first one has to be afferent arteriole. Okay, we are able to get it as afferent arteriole. Cut down the choice. A fine branch of renal artery, afferent arteriole. A double cup-like structure called the Bowman's capsule. Glomerulus along with Bowman's capsule. Malpighian body or renal capsule and proximal convoluted tubule. So this is the choice. Now the proximity between the dash and dash as well as dash in them help in increasing an osmolarity from 300 milliosmoles per liter in the dash and to about 1000 moles per liter in the dash. This gradient is mainly caused by dash and dash. So it is, vasa recta. It is countercurrent mechanism. So this is this enlace there. So you can have cortex, inner medulla, NaCl and urea. So this is what I had explained to you about countercurrent mechanism few minutes before. Small amounts of urea enter the dash of dash which is transported back to the interstitium by the dash. The above described transport of substance facilitated by special arrangement of Henle's law and vasa recta is called countercurrent mechanism that helps to maintain a concentration gradient in the dash. Presence of such interstitial gradient helps in easy passage of water from the dash. So it is going to be, let us look into the choice. It is the thin segment. So you can look for thin segment of the ascending limb of Henle. So it is thin segment of ascending limb of Henle is the first one. Henle's loop. So here it is there. In these two we'll have to look in now. Next is the collecting tubule. Collecting tubule. Inner medullary interstitium, yes, and then it is the collecting tubule, not vasa recta. So B is the answer. Increase in the amount of ammonia in the blood leads to hyperammonemia. Hyper is always more, hypo is less. Renal calculi are the stones or insoluble mass of crystallite salts formed within the kidney. Yes. Glomerulonephritis is the inflammation of glomeruli of kidney. Regarding 1 and 2, both are correct. Regarding excretion of waste products by organs other than kidney, choose the correct option. So all the above are correct. Which of the following is not the kidney? Kidneys are situated below the levels of the last thoracic and fourth lumbar vertebrae, close to the dorsal inner wall of the abdominal cavity is wrong. The flow of filtrate in two limbs of Henle's loop is in opposite direction and thus a countercurrent. That is true. But the flow of blood through the two limbs of vasa recta is not in countercurrent. This is wrong. Even there it is in countercurrent. So correct, but 2 is incorrect. Regarding ANF mechanism, so here all of the above are correct regarding ANF mechanism. Yeah, it has gone to 13th question. GJA or GA, that is juxtaglomerular apparatus, choose the incorrect option. So none of the above, all are correct here. Regarding reabsorption and tubular secretion in urine formation, choose the incorrect option. Nitrogenous waste are reabsorbed by active proteins by passive transport. Analysis of urine helps in clinical diagnosis of many metabolic disorders as well as malfunctioning of kidney. Presence of glucose in urine is indicative of diabetes mellitus. It is true. Presence of ketones in urine is indicative of diabetes insipidus. It is wrong. So 1 is correct, but 2 is incorrect. So I am ending the test. So all the answers are correct. If any answer is wrong, if it will be indicated by red, suppose if this is wrong, you will get a red legend. Then once when you click on that, you will come to know your answer and also the correct answer will be displayed. From this, you can know which is correct. If you have not attempted a question, it will be shown in an yellow color. That means to say that you can still click on that and know the answer. Since it is a negative correction is in progress in NEET exams, you need to understand that if you don't know the answer, don't simply take whatever the answer you feel might be correct. Unless you are sure of the answer or you have a logical way of deducing the answer, please go and pick the answer. Otherwise, don't do. Other you will be losing -1 from your total score. So, with this, we come to the end of today's class. Note that now if you look at NEET full test, we have 38 full hour tests with 15 original tests and 20 plus past NEET papers. All these are solved and they are given with complete explanations. You can also get additional help if you want by using our Ask feature on our site, and we also support chat group using Telegram. If you want to take any one-to-one tutoring from the tutors for any one particular lesson in either of the subjects, you can always contact the tutors. For any inquiries, you can mail to info@clay6.com. So this completes today's lesson. Thank you.